In this video, we are going to look at fatty acid metabolism. We will look at the synthesis pathway, and we also look at the degradation pathway of fatty acids. We will also look at ketone bodies, how they are made, and then how they are used. If you haven't subscribed yet to our channel, please take some time to click the subscription button and also click the bell. And in that way, anytime we post a video, you will all be notified. Also, if you haven't watched the previous video, I want you to know that we started the biochemistry review for the first from the first aid USMLE, and we started from the beginning of the page, and this is almost the end of it. So if you haven't watched the previous video, it will interest you to look at that also. In this video, during the review, I want you to stay to the end, because I will talk about the high yield information that you need, how to approach questions, for the USMLE. I need to turn off this. Good. So now, this fatty acid metabolism, and it has been simplified here. We can see there's a synthesis pathway, and then this is the degradation pathway. So before fatty acids are made, we have to know that this is not in isolation. So if I want to go to the previous sessions, you realize that in the common pathway of the TCA cycle, you can see that fatty acids can be made from a lot of substrate. For example, we can use glucose in the form of ketones. If you want to open to page 74, so that it makes it easier, so that you don't learn in isolation, because I said we have to learn biochemistry as what as a cluster of all the things that are happening together. We don't learn them in isolation. So if I want to look at page 74, that would help us to appreciate this very well. So let's look at page 74. So in page 74, this is glycolysis, and then glycolysis. In glycolysis, we break down glucose to make pyruvate, and pyruvate makes oxaloacetate. Oxaloacetate can be used to make citrate, and our citrate will go into the Krebs cycle and then become oxaloacetate, and then the cycle continues. Now, if there is excess energy, for example, when we just eat and then we are full and we don't need a lot of energy, you finish eating and you go and sleep, and then you don't need a lot of energy, we don't need to be making a lot of ATP, so this cycle will slow down. When it slows down, now citrate will accumulate, everything will accumulate, citrate accumulate, and now acetyl-CoA accumulates. This acetyl-CoA can be used to make malonyl-CoA, and the malonyl-CoA will be used to make fatty acid, and then we can use it later to over here to during metabolism. So this is how we make the fatty acid from glucose. Now, again, we can use all these over here. If you can see all these over here, all this, when we break this into this and this and this, they also go into the TCA cycle, and if the cycle slows, we'll get more of citrate, and then that will go inside the TCA cycle. The same way when you have amino acids, when we have amino acids, some amino acids like glutamate or glutamine can be used to make alpha keto acids or alpha ketoglutarate, and then that will also go into the cycle and make more citrate, and we can use that for in the TCA cycle. So this can be studied in all together. We need to study all of them together. So let's go back to our what we are doing for today. That's page 89, and then we can continue from there. Now, I want you to understand that in fatty acid synthesis, there are two pathways. The first one is the normal pathway, and the second one is the de novo pathway. The de novo is the new pathway, in that in the normal pathway, just when we eat fatty acids in our diet, like the fat that we eat. When we eat it, it just goes straight and then it gets absorbed as fat. We did not really synthesize it, we just digested and then absorbed. But then when in the de novo synthesis, we are using things that are not related to fat, like we are using glucose, which has no connection with fat, to make fat. That is de novo synthesis. Also, we have things like amino acid. If you're using amino acids to make fat, it's also called the de novo, because amino acids have nothing in common with fat. So this is de novo synthesis. That's the meaning of de novo synthesis. So in the synthesis pathway, let's begin from here. The citrate that I showed you, this is the citrate. This is a citrate. And now citrate, this side is in the mitochondria, this is the cytosol over here. So good. So first of all, in the mitochondria, fatty acid needs to come out because the synthesis occurs in the cytosol. That's one thing you want to know, is high yield information. Fatty acid synthesis occurs in the cytosol. So citrate has to come out, but citrate cannot just come out by itself, so it needs a shuttle called a citrate shuttle. So this will be a protein that helps the citrate to leak out of the mitochondria into the cytoplasm. Now, when citrate comes out, we need to use citrate to make acetyl-CoA using the ATP citrate lyase. So it splits the citrate into, or it lyses the citrate into acetyl-CoA. Now acetyl-CoA will be used to make malonyl-CoA. I told you earlier that acetyl-CoA has what? 2 carbons. Malonyl-CoA has 3 carbons. Therefore, it means that we need an enzyme that can do carboxylation. We'll add an extra carbon, that is acetyl-CoA carboxylase. That's how come the enzyme is named this way, acetyl-CoA. We are carboxylating it, so it's acetyl-CoA carboxylase. And now the malonyl-CoA will now be used to make palmitate, which is the 16 carbon fatty acids. Now this is what we have to know. There is a pathway for how malonyl-CoA will be adding things to it, like adding acetyl-CoA in different sequences, but we don't really need that for the USMLE. It's not tested. Yeah, so just know that malonyl-CoA is used to make fatty acid. Now let's look at the regulation of this. Now when we have insulin, excess insulin can stimulate acetyl-CoA, in that insulin is helping to throw more glucose into the cell, right? Meaning that if we can use glucose to make the citrate, then insulin throwing more glucose into the cell means that insulin is helping us to absorb glucose, and we can use that glucose substrate to make fatty acid. But then in the hunger state, glucagon will be released, and when glucagon is released, we don't want to be using the glucose to make fat. We are wasting it. We have to rather inhibit it so that the glucose can stay, and then we can use it to make to make energy for other cells. So this is glucose over here, glucose making citrate, then citrate leaks out of the cytoplasm, right? So it's glucose from glycolysis to acetyl-CoA, then finally everything slows down to citrate. Then we shuttle the citrate out to make acetyl-CoA back. Then this acetyl-CoA will now be used to make malonyl-CoA to fatty acid. So insulin stimulates it. That is during the hunger state, because I said earlier on that when we eat a lot, fed state favors synthesis of fat, because if you don't eat, why would you make fat? But then hunger state will favor the breakdown of fat to make energy back, to make the acetyl-CoA back for energy. All right, now let's look at the degradation of fat. Let's look at degradation. Or first of all, let's just read this, because this is for this side. The top here is for this side. So if fatty acid synthesis requires transport of citrate from mitochondria to the cytosol, predominantly it occurs in the liver and lactating mammary gland and adipose tissue. So you want to know about this. This is the main one. the liver, and then the adipose tissue. Of course, fat should be made in the fatty cells, right? And a long chain fatty acid degradation requires carnitine. So let's look at that. Let's look at the degradation. After we've made it, we can degrade it. Okay, so now when do we degrade fat? One, when somebody has diabetic ketoacidosis, whereby they cannot metabolize glucose, so they have to metabolize their fat. Two, when you are in severe hunger, let's say somebody has traveled for hiking and then their food got lost, or they got lost and then they've not eaten for let's say 3 days, and typically that's how they present the question. Not eating for 3 days, we'll start using our fats to make ketone bodies for energy, because our glucose is going to be depleted. Good. So these are some of the things that favors the degradation of fat. So now the fatty acid that we made here, we have to add coenzyme A to it, and I said coenzyme A is vitamin B5, right? Remember that, because it's very high yield. Coenzyme A is vitamin B5. So remember, that is what we are using to make the coenzyme A. So fatty acid, we use vitamin B5 to make coenzyme A. Remember that, please. Fatty acid plus coenzyme A will be used to make acyl-CoA. It's no more acid-CoA. The name changes to acyl-CoA because one of the hydrogens will leave. So it will become fatty acyl-CoA using the fatty acyl-CoA synthase or synthetase. Good. Fatty acyl-CoA synthetases. Now, the fatty acyl-CoA it's also in the cytosol, so it needs to come into the mitochondria, because degradation of fatty acids occurs in the mitochondria. So we also use the Carnitine Shuttle. So as soon as you hear carnitine, it's about breakdown. As soon as you hear citrate shuttle, it's about synthesis. So now we use the Carnitine Shuttle, and this carnitine is a protein. It's a protein we have carnitine 1 over here in the outer membrane, and then we have carnitine 2 in the outer membrane. But we don't really, it's not really high yield to know this, but yes, they are two, because we have inner membrane and then we have the outer membrane, we have the inner membrane. So just take note that one is in the outer membrane, the other one is in the inner membrane. So carnitine will help us to bring the fatty acid from the cytosol into the mitochondria. Then the acyl-CoA in the mitochondria will now be used to make acetyl-CoA. So you can see that everywhere acetyl-CoA is very, very important. Acetyl-CoA. So we used to make the acetyl-CoA using this enzyme, which is the acyl-CoA dehydrogenase. So all we are removing is hydrogen. That's all. Just is removing hydrogen. And then if what enzyme do you think will do this? It will be the one of the dehydrogenase complexes, right? And vitamin B1 and even B5 and all those things are the thymine pyrophosphate. Yeah. So now the fatty acyl-CoA will be converted into acetyl-CoA, and then we can use acetyl-CoA to make ketone bodies, and then we can also send it into the TCA cycle to make ATP. So this is the degradation pathway of the fatty acid. And we will, in the next page, we will look at how we use the acyl-CoA, the acetyl-CoA, to make the ketone bodies. Now let's look at high yield information here. They like testing this a lot. You want to understand this. This is very, very, very high yield. Please do understand this. Now let's look at this. In the degradation pathway, we have two things over here. We have the deficiencies that can cause diseases in children, especially newborn babies. So when they have systemic primary carnitine deficiency, look at it, this carnitine here is deficient, so they'll always present with low carnitine. In the question, they'll tell you there is decreased carnitine, so you have to know the enzyme deficient is primary carnitine deficiency. Now there is no cellular uptake of carnitine, so the cells cannot uptake carnitine, so there's no transport of the long chain fatty acids that we make here into the mitochondria. So we cannot transport the fatty acids into the mitochondria. This place is deficient, primary carnitine deficiency. This thing is deficient here, so please take note. Now, this leads to toxic accumulation of long-chain fatty acids in the cytoplasm. So all these fats will accumulate, we can't break them down. This causes weakness, hypotonia, and hypoketotic hypoglycemia. You want, this word is very, very important. So the way they present these conditions is that primarily you should know that anytime there's a fatty acid degradation pathway enzyme deficiency, we cannot make ketone bodies. So the question will always come with a child who has hypoketotic hypoglycemia. The child will have hypoglycemia and decreased ketones. Pay attention to the decreased ketones. Anytime there is decreased ketones, know that the answer will be a problem in this pathway, either here or here. If this side is deficient, we cannot make ketones. If this side is also deficient, we cannot make ketones. So please take note. So that helps you to solve questions on this. So it's either primary carnitine deficiency or medium chain coenzyme A dehydrogenase deficiency, and then we just call this one simply MCAD deficiency. MCAD, just like this, MCAD. So please take notes. MCAD deficiency. Okay. Now let's look at MCAD. They also have dilated cardiomyopathy. Take note. But the things that you want to remember is decreased carnitine, and also hypoketotic hypoglycemia. You want to remember these two. Now, the next thing is that medium chain acyl-CoA dehydrogenase, which is the MCAD deficiency, is the one here. Is this side is deficient. If it's deficient, what will happen? The first thing that will happen is that this fat will accumulate in there, because this side is not there, so this fat will accumulate. If fat accumulates, it will be converted into this information is not in the book, but it's converted to dicarboxylic acid. Remember this, dicarboxylic acid. Dicarboxylic acid. So this information is not in the book here, but remember, because sometimes they bring it a lot. So this fat will be converted into dicarboxylic acid. That is 1. The levels will be high. There will also be accumulation of fatty acids in the mitochondria. And the next thing is that there will be decreased acetyl-CoA, right? Because this cannot continue, so the person will still have ketone bodies, like decreased ketone bodies, though. So they'll still have hypoketotic hypoglycemia, because the TCA cycle can also make our oxalate to do gluconeogenesis. So they also have hypoketotic hypoglycemia. So please take note. So the two of them all have hypoketotic hypoglycemia. Let's read here. So this decrease in the breakdown of fat into acetyl-CoA, there's accumulation of fatty acid carnitine. So fatty acid carnitine, that came here. This fatty acid carnitine, there's accumulation of it in the blood with hypoketotic hypoglycemia. Causes, this whole thing will cause vomiting, lethargy, seizures, coma. They'll have liver dysfunction and hyperammonemia. Can lead to sudden death in infants and children. Treat by avoiding fasting. So they should never fast, because anytime they are fasting, they will need this pathway to survive. So they shouldn't fast, so that they can always depend on glucose. So always make sure there's supply of glucose for them by not fasting. So this pathway, it's this all about it that you need to know. So in this one, there's increased acyl carnitine. Just remember, there's also accumulation of acyl carnitine. So that's the fatty acyl carnitine. All right, now let's go to the next page. So in the next page, we want to look at ketone bodies. Now, earlier on, we had said that the degradation helps us to make ketone bodies. So let's see how we make the ketone bodies. Ketone bodies are, if I want to define ketone bodies, they are just high energy molecules that are made from fat and can be used by the body as readily available energy, because they can be used to make ketone bodies can be used to make acetyl-CoA for us to use in the TCA cycle, and then we can use that to also generate a lot of ATPs. So it occurs in the liver. So in the liver, and then fatty acid and amino acids are metabolized into what? Acetoacetate, beta hydroxybutyrate. So this is what we want to know, to be used by the muscles and brain. This is what we want to know. These are the ketone bodies that are used in metabolism, but there are 3 ketone bodies. One of them is the acetone, the one that they always say acetone breath, the Kussmaul breath, that the acetone breath. So when somebody has Kussmaul respiration, and then they breathe in deep and then they breathe out loud, and then they have a fruity smell. That fruity smell, part of the diabetic ketoacidosis, is due to the acetone. So just take note of that. Now, in prolonged starvation, as I said already, and in diabetic ketoacidosis, oxaloacetate is depleted, because we need glucose to make oxaloacetate, right? So if you don't have the oxaloacetate due to prolonged starvation, then you have depletion of oxaloacetate, right? Then there'll be accumulation of citrate. So now all that pathway would be deficient, right? So if the pathway is deficient, means citrate will be there, but oxaloacetate cannot come to join it, so we cannot do TCA cycle. If we can't do that, then we can't make ATP. Therefore, we have to borrow from this ketone body synthesis. So we will use the fat to come into we use the fat to make the acetyl-CoA back. Now this acetyl-CoA will be used to make hmg-coa. We are just, in this case, in this place, you shouldn't be confused, because in this synthesis, we are just adding more acetyl-CoA. We are adding more acetyl-CoA. We are adding another acetyl-CoA, first of all, to make acetyl-CoA. Then we'll add another one, then we'll get hmg-coa. So it's just, ketone bodies are just like a polymer of acetyl-CoA. We are just adding more acetyl-CoAs together. So if I take the first acetyl-CoA, I'll add a second acetyl-CoA, then that will be called, it's not here, but over here in this place, it will be called acetyl-CoA. You can check that, you can Google it, and then we'll add another acetyl-CoA, then we'll get hmg-coa. hmg-coa will be used to make acetone, the first one. Acetone, the one that we saw here, that's the first ketone bodies. We'll add another, we'll change this acetone, then we'll get beta hydroxybutyrate. So acetone can be used to make beta hydroxybutyrate, and it's reversible. Now, this acetoacetate goes into the blood, and the acetoacetate can be used to make acetone, because the names look alike. The acetone can go into the lungs and be breathed out. There is no way we can measure acetone, because anytime it's made, we can't measure it accurately, because anytime it's made, we breathe some out and we lose it in the air. So we don't use that as a measure of ketoacidosis. Okay, we don't use acetone as a measure of ketoacidosis, but then we can use the acetoacetate, and we can also use the beta hydroxybutyrate. These two can go into the skeletal muscles to be used for energy. So that's what, and then when they get into the skeletal muscle, beta hydroxybutyrate is converted into the acetoacetate, and then this acetoacetate can go into the TCA cycle, and then part of it also become acetyl-CoA, as I said over here, and this will be used to make what? Acetyl-CoA, and then the muscles can use that to make energy. Now, high yield information here is that you have to know that the fruity smell is acetone. That's high yield. 2, urine tests for ketones can detect only these two, acetoacetate and not beta hydroxybutyrate. Okay, so they are saying that the urine test can only detect acetoacetate but not beta hydroxybutyrate. But blood test can detect both of them. That's what you want to know. Blood test can detect both of them, but urine test is only for acetoacetate. So this is only for urine, but both of them can be detected in blood. Right now, remember that all these things will be occurring in the mitochondria. All this will be occurring in the mitochondria. So when we have the ketone bodies, they cannot be used by RBCs because the RBCs do not have mitochondria. So RBCs cannot use ketones. They strictly use glucose because they don't have mitochondria. That is why. So RBCs don't do all these things that we are talking about here. Now they can't synthesize it. Also, they can also metabolize. Okay. Now hmg-coa lyase is for ketone production, but then hmg-coa reductase is for cholesterol synthesis. That's why we give them the statins. The statins block hmg-coa reductase. Statin. So the statins block hmg-coa reductase. Okay, so just take note of this. Anytime you see hmg-coa lyase, because hmg-coa, the lyase is here. Lyase, hmg-coa lyase is breaking this hmg down to make acetoacetate and beta hydroxybutyrate. But then the hmg-coa reductase is used in the cholesterol synthesis. So take note of that. Yeah. So this helps us a lot. Now, another thing that we didn't talk about is that with chronic alcoholism, overuse, like in chronic alcohol overuse, if somebody drinks a lot of alcohol and is chronic, we said that there will be excess NADH, and that excess NADH shunts oxaloacetate into the malate, because NADH means that there is high energy state, so TCA cycle will slow down. We will not need it again because we don't have any. We need NAD plus, which is low energy state, so that the TCA cycle can be running. But if you drink excess alcohol, then we have to break the excess alcohol into the alcohol like in the alcohol metabolism pathway, whereby we said that we'll use alcohol dehydrogenase to break the alcohol down into what did we break it down to? We use let me just refer this quickly. I think that skipped me a little bit. Yeah, we'll break the alcohol down into here it is. So acetaldehyde, correct. We break the alcohol down into acetaldehyde. That's on page 72. So we said we're breaking the alcohol down into acetaldehyde. So in that case, we will make a lot of NADH. So when we have accumulation of NADH, it means that the body will perceive this as high energy state, so the TCA cycle will slow down. If TCA cycle slows down, oxaloacetate will be shunted into the malate to be shunted, like to be converted into malate. And when we get the malate, it means that gluconeogenesis cannot occur. If gluconeogenesis cannot occur, what will happen? We'll rather use our fat to make ketones. That's why alcoholics can also have high ketone bodies in their blood. So please take note of that. So in the next video, we'll start from the fasting and then the fed state, so that we understand how we use energy. All right, so thank you very much for joining me in this session. I wish you the best of luck in your studies. I want you to know that it's doable. Yes, you can. You just need to be consistent and study everything every day, and continue to do your questions. When you get it wrong, come back to the book and then look at why you got it wrong and write your little notes down. And yes, at the end of the day, it would pay off. Patients are waiting for you. America is waiting for you. Your family is waiting on you. So thank you very much. Please subscribe to my channel, and just take time to write a little comment for me, because that will help YouTube to suggest my video to other people. The algorithm detects like the channel is busy, and then they suggest it to other people, and it will reach the whole wide world. Thank you, and good luck in your studies. You can make it. Peace out.